Hello, my Form 5s. How is everyone doing? Okay, sorry for the camera. Okay, welcome back to our weekly class for June week 4. Okay, today we are going into a new chapter. Okay, I hope you're ready. We finished our reproduction. Okay, I would say quite a difficult chapter. Quite a lot of things to memorize. Okay, this chapter 5, there will be much less things to memorize, okay, but it doesn't mean it's easier. Okay, this part is very crucial in your exam, so please pay attention. A lot of, I would say, logical assumption stuff here. Okay, not much things to memorize because this part is more mainly about genetics. Okay, how inheritance happens. Okay, because this chapter is called inheritance and variation. Okay, can you see I combined chapter 5 and 6 together because they are the same thing. Okay, it's about how I pass on something to the next generation. Okay, very simple concept here. Okay, so inheritance and variation. Inheritance, basically I pass something down to the next generation. Variation basically means why are we all different. Okay, basically inheritance that causes variation. Okay, if you put it that way. All right, so let us begin by this Mendel's experiment. Okay, 5.1. Okay, we'll go into the Mendel experiment later. Let's first understand your chromosome. Okay, your chromosome. Okay, so there are a lot of words of here that you should know. Okay, a lot of words here. Okay, might look very hard, okay, but no, it's not hard. Okay, so just imagine this is your homologous chromosome. Okay, what does it mean by homologous chromosome? I'm sure you know this. One side is from your paternal side, your father's side, and then one side is your maternal side, your mother's side. Okay, remember you have 23 pairs of chromosome. Okay, 23 comes from a father, 23 comes from a mother, so we have 23 pairs. Okay, it's like the left side of your shoe and then the right side of your shoe. You have one pair of shoes. All right, so homologous chromosome. Okay, so let's look at the first word, allele. Okay, what is allele? Okay, what is allele? Allele basically, okay, it sounds like parallel. Okay, sort of like parallel. So they are parallel. So allele. Okay, what is allele? Different forms of the same gene for a trait that occupy the same relative position. Okay, so these are a pair of allele. Okay, sorry for that. Let's understand what is gene first. Okay, I skip something. Okay, so what is gene? Gene is the basic unit of inheritance. Okay, you inherit a whole sequence of gene. Okay, if you're still confused, never mind. Okay, imagine this is your chromosome. Okay, this is your chromosome. Chromosome is like a big roll of thread. Okay, a big roll of string. Okay, so if you pull the string out, okay, so the string, if you zoom it inside, it looks like blocks like that. Okay, so each of these blocks we call it DNA. Okay, DNA, each is DNA. So let's say this sequence of DNA, let's say here has 1000 DNA. Okay, and then this 1000 DNA, it determines your height. So this 1000 DNA, we classify them as a gene. It's a unit. This 1000 DNA, you cannot split them up. It means nothing. You have to put them together and it determines your gene. So this is your height gene. Okay, hope I'm making sense here. So this is height gene. So this sequence of DNA determines your height. That's a height gene. Okay, so if your dad passed the height gene to you, he will pass on the 1000 unit to you, okay, as a unit. It's not like he'll pass half to you. It doesn't work that way, okay, because that 1000 unit determines a certain setting. Okay, so we have height, you have eye color, okay, skin color, hair texture. Okay, we'll go into that in the next coming lessons. Okay, so you have to know this just for now. That's your gene. So basic unit of inheritance. Okay, so can you see this is one gene from your father, let's say, and then one gene from your mother. So they are the same. Let's say this is height, and this will also be height. Okay, you will not have height here and weight here. It doesn't happen like this. They have to compare the same thing. Okay, so can you see they are the same? They are the genes that determine the same characteristic. Okay, so we don't say characteristic here, we say trait. Okay, so trait is characteristic. Okay, basically the same thing. Okay, so can you see they determine the same trait, so we call them allele, as in parallel. Okay, allele, parallel. Okay, the name is almost the same. Okay, parallel. And then at this location, This location, we call it locus. Locus, location. Locus, location. So the locus, it's there. Okay, the position of the gene. Okay, and then okay, so these five are together. Okay, 1, 2, 3, 4, 5. Okay, so it's not actually that much difficult. Okay, so what is dominant allele? Okay, dominant allele is sort of like the winning guy. Okay, if dominant is present, it will always show. Okay, what do I mean by this? Okay, let's look at the point. Okay, allele that were expressed in an organism's phenotype and mask the effect of recessive. Okay, you don't understand. Okay, never mind. Let's say, okay, for dominant allele, we always put alphabet capital letter because it's dominant, it's winning. Okay, so let's say now for height, you have two genes here. You have two types of gene. Number one is big T. This is tall. Okay, small t, which is short. Okay, so who's the winning guy? Tall is the winning guy. Which means if your father gives you a tall gene, the winning guy, and your mom gives you a short gene, who will win? The tall guy will win. So you will be tall. Okay, the short gene will not be expressed because it's the losing guy. That's why we call them dominant allele. Okay, dominant will always be shown. Always. Always. Okay, so as the name says, recessive allele. Okay, it's the losing guy. Okay, so what does it say here? Allele that will only be expressed in the absence of dominant allele. Only if there's no dominant, recessive can show. Okay, let's say your dad gives you a short gene, your mom gives you a short gene. Okay, there's no dominant here, no winning guy here, then of course you will be short. Okay, if there's one winning guy, okay, no matter one or two, you will still be tall. Okay, if you have one big T or you have two big T's, you will still be tall. Okay, so it's represented by a small letter. Okay, so what is phenotype here? I'll mention that later. Okay, so from this, we can know that you have three combinations here. Okay, either you can be big T, or you can be big T, small t, or you can be small t. Okay, we have three combinations here from these two genes. Okay, so these three, we classify them as here. Okay, so can you see both of these? They are the same. Okay, big T, small t. So same, we call them homozygous. Okay, as the name says here, homozygous. Okay, what does it mean by homo? Homo means same. Homogeneous, homosexual. Okay, same sex marriage. Okay, homosexual. So homo is the same. Okay, so homosexual. So you have two types here. Can you see here? Homozygous dominant, which is the winning one, which is two big T lah. Okay, and then you have homozygous recessive, which is the two losing guys. Okay, so we have two small t's here. Very simple. And then for the middle guy, okay, can you see they are different? So you have homosexual, and then for guys and girls one, we call it heterosexual. So it's mentioned here, heterozygous. Okay, can you see I don't put dominant or recessive here because there's no two types. Heterozygous dominant or heterozygous recessive, they represent the same thing. As long as it's one big, one small, it's heterozygous. Okay, so like I said, this part is not so difficult. Okay, mainly it's about common sense stuff. So an individual who has pair of identical dominant allele, okay, identical, they have the same tall gene, okay, like let's say MM here, and then heterozygous has two different allele, homozygous has identical, and then it's recessive gene. Okay, so these two are the same thing, only one is dominant, one is recessive. Okay, done. Okay, and then what is genotype and phenotype? Very simple. Okay, genotype is your gene. Okay, the combination of gene. So let's say just now, in terms of if I use R, okay, you have big R, small r, small r. This is the genotype. Phenotype is what you can see. Okay, so let's say now, okay, for example, big T is tall, big T, small t still tall because I have a winning guy. Okay, and then small t, I'm short. Okay, this is the genotype, and then this is the phenotype. Okay, so can you see big T and big T, small t? They are different genotype, but they are same phenotype. Okay, So at the end of the day, we only care about phenotype, okay, because that's what you can see. Okay, that's what you can see. Okay, so accessory here says, can you see big R, red flower, assuming red is dominant. Okay, big R, red flower, small r, white flower. Can you see different genotype here, but same phenotype here? Okay, so if you are tall, it could be you being big T or big T small t. Okay, although maybe you and your friend, you have the same phenotype, both of you are tall, but maybe genetically you are different. Your genotype is different. How would you know? You need to go and do a DNA test. There's no way for you to know besides that. Okay, so okay, doesn't matter. Okay, so we have dominant trait here, recessive trait here. Okay, like I mentioned just now. Okay, so let's look at Mendel's experiment. Okay, please bear with me, the video will be quite long because quite a very important concept here. I would say this part is like 70% of this chapter. Okay, so Gregor Mendel, okay, who is he? He is a bishop. He was a bishop. Okay, a bishop as in a church worker. Okay, he's the first guy who studied genetics, inheritance. Okay, so we call him the father of genetics or the father of inheritance. Okay, because since the beginning of times, we somehow know, although it's not very good, we somehow know that we will somehow look like our father and mother. Not so much as our grandfather and grandmother, somehow we look like father and mother. Okay, we know this will happen, but we don't know how. Okay, so he's the first guy that proposes the mechanism of how everything works. Okay, very interestingly. Okay, so study of inheritance, okay, passing on genetic information from one generation to the next generation. Okay, discovered by Gregor Mendel. Okay, so so you have so many characteristics to study in a plant. Okay, like us humans, how many characteristics we have? Thousands. Okay, thousands. Okay, so for pea plants, not as much as us, but same thing. Okay, like seed shape, you have round and wrinkled. Seed color, you have yellow and green. Flower color, you have purple and white. Pod shape, inflated and constricted. Pod color, you have green and yellow. Okay, flower position, you have axial and terminal. Stem length, you have tall and short. Okay, but for Gregor Mendel, he kept it simple. Okay, he kept it simple. Okay, he did something called monohybrid cross. Okay, monohybrid cross. What does it mean by monohybrid? Mono means one. Okay, mono means one. Because he knew that it is impossible to study all these characteristics. Okay, so he just studied one. Okay, because basically if one works, the rest will work the same. Okay, so he just studied one trait. Okay, let's say now if, okay, for example, okay, in this case he studied seed color. If I'm not mistaken, he studies seed color. Okay, so if I'm studying seed color, let's say if I'm Gregor Mendel, I'm studying seed color, I don't care about other traits. So I don't look at the seed shape, flower color, pot shape, pot color, flower position, stem. I don't care. I am only looking at the seed color. Let's say I'm studying human, about height. I only care about the height. I don't care about the skin color, hair texture, okay, earlobe, fingerprint, whatever. I don't care. I'm only studying the height. Okay, of course, why did he use human? Human is super hard. Okay, it's not very ethical to do experiments on human, and of course, it will take very long. Okay, for a human to grow up, it takes years. Okay, but for pea plant, it's only months. Okay, few months I can really see their characteristics. Okay, I can study them. Okay, so this is what we call Gregor Mendel's first law. So this one you should memorize. Okay, Mendel's first law is the law of segregation. Okay, that's the name of the law you have to know. This is very commonly asked in objective question. Okay, what does it mean by segregation? Segregate means that separate. Okay, separate. Okay, so what does it mean by here? Okay, let's say now, okay, basically the law of segregation is like this. Okay, let's say your father is like this. Your father is tall. Okay, but he has a short gene. Okay, maybe he got it from your grandma, and then your grandpa is tall. So your father is like this, big T, small t. And then when he do meiosis, okay, remember your father only gives you one. Okay, he won't give you two. You won't take two. Then if you get another two from your mother, you will have four. That is, okay, you're not human anymore. Okay, so half of his sperm, okay, 50% of his sperm is tall gene, 50% of his sperm is short gene. Okay, that's why if your father is tall, okay, if you are short, you got his short gene. He is a heterozygous. I hope I'm making sense here. Okay, so his sperm is 50% tall gene, 50% short gene. Can you see? They are separated. Okay, the sperm is 50%, 50% separated. That's why Mendel called it the law of segregation. Okay, the law of segregation. All right. So what does it say? The members of each pair of alleles separate or segregate, okay, during the formation of gametes. Okay, like here. Okay, I segregate them by meiosis. Okay, meiosis is to give you half set of chromosome. Okay, only one allele can be carried by a single gamete. Okay, the gamete, which is the sperm or the ovum, can only carry half. Okay, you are a full, you have 46. Your father can only give you 23. Your mother can only give you 23. Okay, so it's a random thing. Okay, so like in metaphase 1, okay, I'm sure you have learned this. This is meiosis, in meiosis 1. Okay, so now you have big T and short t here, big T, small t. Okay, so chromosome is moving towards opposite pole, LLT is separated. Okay, so that's why you separate. Okay, so your father separates, your father might give you this or it might give you this. So 50-50 lah. Okay, maybe your brother is tall, then you are short, then you got the short change, you got short change, then your brother got lucky lah. Okay, this is genetic, you can't blame anyone. It's like playing a coin flip or flipping a coin, or you know, throwing a dice. This is a chance game. Okay, so monohybrid cross, like I said, it involves a pair of allele cross between a tall. Okay, he did this experiment by doing a tall and short pea plant. Okay, so monohybrid inheritance means a single characteristic only. Okay, if they ask this in an exam, what does it mean by monohybrid cross? It means it only involves one characteristic, because it is impossible for us to study all. Okay, there will be too many. Okay, so what did he do? Okay, so he took a pure breed tall plant. What does it mean by pure breed? Okay, so tall plant can be big T or big T small t. Okay, pure breed means this. Okay, pure breed. Okay, how did he know this is pure breed or not pure breed? You don't ask me. That is his problem. Okay, all you have to know is he used a, let's just assume he knew. Okay, so he took a pure breed tall plant. Okay, a pure breed tall plant, and then he took a dwarf plant, a short plant. Short plant is confirmed small t, small t. Okay, because if it's big T, small t, the plant will be tall. I have a dominant gene there. Okay, a plant will only be short if it has a 2 recessive gene, both also bad gene. Okay, only that can show. Okay, so please learn how to draw this. Okay, in SPM they ask this all the time, and it's easy to score. Okay, you don't need to write explanation. Okay, long without explanation, you just have to draw this. Okay, so tall plant. Okay, if let's say your daddy is tall, okay, pure breed tall. What does it mean by pure breed? Your grandfather, your grandmother, your great-grandfather, your great-grandmother, everyone is tall. Everyone from your father's side is super tall. Means most likely 99% he'll be pure breed. Okay, so in his sperm, okay, so he does meiosis. In his sperm, it will be 100% tall gene. Okay, he doesn't have a short gene because he's pure breed. A short gene doesn't exist in his family. You know what? Good for him. Okay, so that's what we call you win the genetic lottery, because you're super lucky. Okay, so let's say now your mom is short. Everyone in the family is short. So your mom is confirmed short t, okay, small t, small t. So in her ovum, everything will be short t, because in her gene, there's no big T. Big T doesn't exist in her family. Okay, so when you cross them together, okay, I don't like to draw it like this because it's too simple. Okay, I like to cross it like this. Okay, so 1, 3, okay, 1, 4, okay, and then 2, 3, 2, 4. Okay, remember 1, 3, 1, 4, 2, 3, 2, 4. So the first child, okay, can you see F1 is first generation. Genotype is what you see, what's the gene. Okay, so the first one is big T, small t. Can you see big T here? Okay, big T here, and then 1. This is 1, and then 3 is this, and then short t. Okay, big small. Okay, because big T and small t here, big small. So they simplify by just writing 1. Okay, for me, I like to write all. Okay, because it's less confusing. Okay, so big T, small t is tall. Okay, the phenotype, what you can see is tall. They're all tall. Okay, so this is what he did, the first generation. So from this experiment, okay, the first generation he planted, almost 99% of them are tall. So from this experiment, he knew that you know, the dominant one will always win. Okay, that's, you see, last time nobody knows this. He's the first guy who discovered this, and he sort of back engineered this concept. Okay, so quite amazing of him to do this. Okay, so and then what he did was he used two first generation, and then he crossed them again. Okay, he crossed them again. Okay, so tall plant, okay, I crossed this and this, okay, because this is the F1 generation. Okay, now different story. If your dad is not pure breed one, your dad will be, the sperm of your dad is 50% tall, 50% short. So we should see which one will kena lor. Okay, and then your mom is the same thing, 50% tall, 50% short. Okay, so when I cross them together, 1, 3, 1, 4, 2, 3, 2, 4. Okay, so the first child, okay, the first possibility is 2 big T's. Second possibility, big small. Third possibility, big small. Fourth possibility, small small. Okay, so you sort, okay, this is tall. 2 big T's are tall. 1 big, 1 small, still tall. 1 big, 1 small, still tall. 2 small T's, it's definitely dwarf or short. Okay, so I get a ratio of 3 to 1. So in his second generation, he got 3 tall plants and 1 short plant. Okay, so from, imagine now I cover this right side. He only did the left hand side experiment. Okay, this one. He did all this, and then he made a conclusion of this. That's why we call him the father of inheritance, because that is pretty amazing. Okay, for someone that can, you know, make this assumption, make this hypothesis, which is proven to be a theory today, it's pretty damn amazing. Okay, that's what Mendel did. All right, and then he did another experiment. Okay, so remember just now I used one characteristic, which is tall or short, the height of pea plant. Now I use 2. Okay, so just now the first experiment, he didn't care about anything else besides the height. He didn't care about the seed color, the seed texture, the blah blah blah, okay, the other characteristics we mentioned just now. Okay, he did not care. Okay, so now he did an experiment, the second experiment. Okay, the second law of Mendel's, okay, the name of Mendel's second law is called the law of independent assortment. Okay, please remember this. Okay, what does it mean by independent assortment? Independent means I don't rely on you. Let's say height gene and seed color gene, okay, or seed texture gene, are they relying on each other? No. How tall am I has nothing to do with the seed color. That is another story. That's why we call them independent assortment. You get what I'm trying to say? Okay, so let's say you are tall, you have double eyelid. Maybe your brother is short, but he still has double eyelid. Okay, but the double eyelid has nothing to do with your height, because that's a different gene. Okay, so this is what is meant by second law. Okay, which means one gene doesn't affect the other gene. Okay, one trait of gene of one trait doesn't affect the gene of another trait. Okay, because they are controlled by another different set of gene. Okay, so here he used 2 characteristics, so it will get a bit more complicated here. So 2 or more pairs of alleles segregate independently of one another during formation of gametes. Okay, can you see? Segregate independently. It means they're not dependent on each other. Okay, so possibility 1. Okay, so can you see here? So now let's say I use height gene, okay, and seed texture. Okay, this is what he did. Okay, so can you see? This is the tall gene, short gene. Big R is round seed, short R is wrinkle seed. Okay, so when he sort them, okay, this is what meiosis does. So can you see? This is the first combination, dominant, 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 dominant. Second combination, recessive, 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 recessive. Okay, and then for this one, you have different more combination, short R, big T, short R, big T, big R, small t, big R, small t. Okay, so can you see? The R doesn't affect the T. Okay, so T is height gene, R is seed texture gene. They are not dependent on each other. Okay, so very simple. So this is what we call a dihybrid cross. Just now it's monohybrid. Mono is 1. Okay, di is 2. So dihybrid cross. Okay, so dihybrid inheritance. Okay, so this one is a bit more confusing. Okay, so this time he used yellow and round seed, okay, because it's easy to see. Okay, so yellow and round seed. Yellow pure breed means both big Y lah. Okay, and then round pure breed round seed means both are. Okay, means everything is dominant here. This is the perfect gene. And then this one is all the weak ass gene. So you have small r, and small y, okay, which means they are green and wrinkled. Okay, so green being recessive, wrinkled being recessive. Okay, so when you cross them to, okay, this is a bit confusing now. Okay, just now, for big T, big small t, you have only two combinations, which is one big T, one small t. Sperm has 50%, 50%. Okay, but in this case here, it's different. Okay, so okay, the first generation, okay, all the gene will look like this. Okay, all the gene will look like this. Okay, because this seed has only big Y and big R. Okay, so all the sperm or ovum, the gamete inside has only big R and small r gene. So I don't need to, okay, if you want, you can write it 4 times. Okay, but it's quite unnecessary because they're the same thing. Okay, and then for the recessive one, the weak ass one, okay, all will be small. Okay, because they only have recessive gene. Okay, so when you cross them, okay, since everything is the same, I will get all the same children. Okay, which means I will get big Y, small y, big R, small r. Okay, you have a dominant Y here, you will still be yellow. You have a dominant R here, you will still be round. Okay, the problem comes is, okay, this is F1 generation. The problem comes during F2 generation. So again, he did the same thing. He used 2 F1 generation and he crossed them again. Okay, so now it's more confusing. Can you see I have 4 combinations of gametes now? I have big Y, big R. Okay, because I have 4 types of gene here. I have big Y, big R, small r. We are only talking about 2 characteristics here. We already have 4 different combinations. Can you imagine us human? We have thousands of genes. So every sperm will be different. Very different. That's why you will never get you'll never get 2 same children. It's impossible to do so. Okay, because 10 millions of sperm, 10 million of them are different. Okay, so you have 4 combinations here. Okay, big Y, big R, big Y, small r, big, okay, small, okay, I'm having a tongue twister here. Okay, so small y, big R, small y, small r. Okay, so 4 combinations here. Same case for the second guy. Okay, big, I just want to say big, okay, big, small, big, small. Okay, so now it's not the case of just doing 1 3 1 4 or 2 3 2 4. Okay, you can cross them. This will be super confusing. It's like 1 1 1 2 1 3 1 4 2 1 2 3. Okay? By the time I do this, you will die of confusion. Okay, instead we draw something called a Punnett square. Okay, we draw a Punnett square. So we draw all the male ones here. Okay, the all 4 combinations here, and then the female 4 combinations here. Okay, so we cross them together. So the first, okay, you can read it yourself lah. The first one, okay, all dominant, so round yellow. Okay, second one still round yellow. Okay, because yes, I have a small r here, but I still have a big R here, so I'll still be round. Okay, so calculate everything, you will get a ratio of 9 3 3 1. Okay, 9 3 3 1 means I have 9 yellow round, 3 green round, 3 yellow wrinkle, 1 green wrinkle. Okay, this experiment shows that the color of the seed has nothing to do with the texture of the seed. They are controlled by 2 different genes. Okay, which means your height gene will not affect your eye color gene. They are determined by different genes. Okay, so they will not affect. That's why we call it independent assortment. Okay, this is what Mendel said. Okay, so that's it for this week. Okay, I'm gonna stop here. We will go into some exercise. Okay, let me emphasize yet again, this is the main part of this chapter. If you can't understand this, you will not understand next week, because we are doing the same thing all over again, which is a crossing 1 3 1 4 2 3 2 4. Okay, so basically this crossing thing, it's this whole chapter. If you don't understand this, basically you don't understand the whole chapter. Okay, so please waste some time in understanding this. I wouldn't call wasting, you just use some time. Okay, so anyway, let's look at the exercise. Okay, so Mendel first and second law, schematic diagram, monohybrid cross. Okay, B represents curly hair. Okay, so big B, curly hair is dominant. Okay, and then straight hair is recessive. So I have a recessive gene because I'm straight hair. Okay, so phenotype. Okay, both, it says here both homozygous. Okay, so curly hair will confirm be B. Okay, straight hair confirm is too small b. I cannot be like this, because if I'm like this, I will be curly hair. Okay, so no. Okay, so the gametes here, if your daddy is pure breed curly hair, all his sperm will be curly hair. Your mother, of course, straight hair will be all recessive gene lah. Okay, so when you cross them, 1, 3, 1, 4, 2, 3, 2, 4. So you get big, small, big, small, big, small, big, small. So this is your genotype. And then for the phenotype, this is curly, okay, curly, curly, curly. Okay, so genotypic ratio, it's 4 to 0. Okay, phenotypic ratio, it's also 4 to 0. Okay, all the same thing. Okay, and then for heterozygous, okay, can you see here both heterozygous? Oh, I forgot to mention something here. This is BB, this is BB. Okay, so since both heterozygous means big, small, and big, small. Okay, so your mommy will be curly hair, your daddy will be curly hair, but they are not pure gene curly hair. They have a recessive gene hidden there. Okay, so for your daddy, the sperm, half is curly hair, half is straight. Same as your mommy's ovum, half curly, half straight. Okay, so when you cross them together, 1, 3, 1, 4. Okay, 2, 3, 2, 4. Okay, so the first one will be big, two big ones. Okay, and then big, small, small, small. Okay, so genotype is curly, 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 straight. Okay, so the genotypic ratio, very similarly they ask this, is 1, 2, 1. I have 1, 2, 1. Okay, and then for phenotypic ratio is 3 to 1. 3 curly and 1 straight. Okay, so as long as you can complete this diagram, you already understand this chapter. Okay, so you might say, hey sir, how about the dihybrid cross one? The one I would, if you cannot understand that one, you don't have to. Okay, you just need to know that it's dihybrid cross and then you have the Punnett square. That is just a more complicated version of this. Okay, so one of the curly hair is homozygous, another curly hair is heterozygous. Okay, so okay, so let's say father is homozygous curly lah. Okay, so your father has pure gene. Okay, your mother is heterozygous, so your mother is like this. Okay, doesn't matter father or mother lah, okay, same thing lah. Okay, so the gamete will be all dominant gene, and then for a mother will be half dominant, half recessive. So again, 1, 3, 1, 4, 2, 3, 2, 4. Okay, so we get big, 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 small, big, 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 small. Okay, so it will be all curly, 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 curly. Okay, so genotypic ratio is 1 to 1 because you have 2 big B's, okay, and then 2 big small. Okay, and then phenotypic ratio is 4 to 0. Okay, so very good practice here because this is basic. Okay, I hope you can do all this. Okay, so curly hair is heterozygous, and then straight hair. Okay, so curly hair is heterozygous, confirm is big, small. Straight hair, you don't need to ask question, confirm is small, small. Okay, so gamete again, the father will be half dominant, half recessive. Mother will be all recessive. So when you cross them, 1, 3, 1, 4, 2, 3, 2, 4. So you get big, small, small, small. Oh, sorry. Okay, big, small, 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 and small, small. Okay, so phenotype here is 2 curly, okay, and 2 straight. Okay, genotypic ratio is 1 to 1. Phenotypic ratio is also 1 to 1. Okay, so we have 4 different scenarios here. Okay, so first scenario is your father being very perfect, your mom is no perfect gene, okay, not perfect or all recessive gene. Second case is your father and mother same. Okay, they are not the pure breed one. Okay, and then third case is your father is very perfect, your mother is half perfect. Okay, so and then fourth case is your father is half perfect, your mother is not perfect at all. Okay, no dominant gene. Okay, so 4 scenarios here. Okay, as long as you can understand all these 4 scenarios, you can understand everything in the next lessons. Okay, we will study about blood type, we will study about gender. Okay, so make sure you understand this. This is, I can't stress this enough, this is the main part of this chapter. Okay, so dihybrid cross. Okay, this is what we dread to see. Okay, so let's say Yutian is tall but ugly, wife is dwarf but good looking. Okay, if both are homozygous, state the possibility F1, F2 generation. Okay, so let's just imagine, okay, he's tall, okay, both are homozygous, which means he will be big T, okay, but ugly. Okay, there's no gene for ugly, but this is just an assumption, okay, because ugly or not, okay, ugly is dominant. Would you look at that? Okay, so ugly means big H, big H. Okay, the wife is dwarf. Dwarf means small t, but it's good looking. Since they say it's homozygous, so good looking is small h, small h. Okay, so for daddy, okay, and then for mommy, daddy is big T, big H, big H. Mommy is small. Okay, so for this one, you don't need to confuse yourself. There's only one type of gamete possible, which is big T, big H, because there's small here. So for this one is small t, small h. There's only one possibility. Okay, so when you cross them, the children will confirm be big, small. So the children will be tall and ugly. Okay, so ugly or not ugly, this is in the eye of the beholder lah. Okay, some say single eyelid is ugly, some say it's not ugly. Okay, it's a very subjective thing. Okay, anyway, so we still have a bit of time. Okay, I will try to finish this. Okay, so let's look at question 1. Okay, so shows a cross between heterozygous black. Okay, heterozygous means big, small lah. Okay, and a homozygous white, which means small. Okay, so black fur is dominant, and then white fur is recessive. Okay, so you know the male is big, small, the female is small. Okay, so the sperm will be half big, half small, the ovum will be all small. Okay, so when you cross them together, this is big, small, small. Okay, like one of our 4 scenarios up there. Okay, this applies to sheep. Same thing. Okay, it's the same thing. Okay, so complete diagram 1 by filling the genotype of the parents. Gamete they produce and genotype. Okay, we've done this. You see, 4 marks. Okay, so this chapter is super easy for you to score. Which of the offspring labeled in F1 are heterozygous and homozygous? Okay, so who is hetero? Hetero, we have P and Q because they are different. Okay, big, small. Homo is R and S. Okay, so would the result in F1 generation be the same if parents were heterozygous black female and white male? Okay, means now mother is like big, small, daddy is small. Will it be the same? Yes, same. Okay, because same genetic composition, okay, or gene in parents. Okay, yes, they switch gender, but the genetic composition is still the same. Okay, so produces children or offspring with identical gene. Okay, which means same as this. Okay, so produces same result. So doesn't care male or female, as long as the gene is the same, I will produce the same thing. Okay, so C, if a homozygous black male, which is big, and heterozygous black male are crossed with heterozygous is big, small, crossed with heterozygous black female, so big, small, what is the ratio that will be obtained? Okay, Oh, so means the female bred two times. Okay, so big small times big small. Okay, so you can just do it the 1 3 1 4 yourself lah. Okay, so by looking at this, you will get big small. Okay, big small. For this one is big small, big small, small. Okay, so what's the ratio here? Okay, this is black, so 4 to 0. Okay, black, white, 3 to 1. Okay, so let's look at one more question. Okay, a farmer has 3 types of plant, Q, P, and S. Q and P produces red flower, S produces white flower. By using a small brush, the farmers carry out pollination between Q and S. Okay, so and between S and P. The crossbreeding between Q and S produces plant that only have red flower. Crossbreeding between SP produces both white and red flowers. Okay, which of the following 3 parent plant is heterozygous? Okay, not heterozygote, heterozygous. Okay, so okay, it looks very confusing here. Okay, so what they did was, okay, so let's just assume, okay, they should mention which is dominant. Okay, it could be white dominant or red dominant. Okay, but from our understanding, red is dominant. Okay, so okay, this question is a bit confusing. Okay, so Q and P produces red flower. Okay, so Q and P is dominant. Okay, right, dom. Okay, S is recessive. Okay, assuming white is recessive. Okay, of course. And then when I cross Q and S, dominant and recessive. Okay, and then I cross S and P, which is also dominant and recessive. Okay, so Q and S produces only red flower. So I know that Q is a homozygous dominant, because Q is very dominant. All huge letter, all large letter. Okay, all big letters. Okay, so and then so it will produce all. I hope you understand what I'm trying to say here, which means Q. Okay, the gene of Q is RR. Okay, S is small r. So the offspring will be all like this. Okay, so all the offspring will be red flower. Okay, that's what they are trying to say. So SP produces white and red flower, which means P is hetero, which means P is like this. Okay, and then S is like this. So we get big small, small, and small. So P is not the pure breed one. Okay, so who is hetero? Of course, is P. So draw genetic diagram to show S and P. Okay, so basically this one. Okay, drew this. Okay, what is the phenotypic ratio of F1 generation? So it's 1 to 1. Okay, write the genotype for plant S. Okay, of course, it's small. What is the name of the plant S in terms of genetic content? So what do you call this? This is homozygous recessive. Okay, so D, if the product of the crossbreeding between was it Q, or this is Q, my mistake here, and S is crossed among themselves, okay, what is the genotypic ratio, phenotypic ratio for the plant that is produced? Okay, so so Q and S, I produce this. Okay, so now I cross them again, which is the second generation experiment lah. Okay, so I get big, small, big, small, and small. Okay, so genotypic ratio is 1 to 1. Phenotypic ratio is 3 to 1. Okay, so draw a Punnett square. Okay, this is not dihybrid cross. You don't have to draw a Punnett square. Since they ask for it, you just draw one. Okay, so for the, okay, draw it like this. Okay, this is the male, this is the female. Okay, so you have R and R here. Okay, same thing, you have R and R here. So this is big, small, small. All right, pretty simple. What is the name given to the type of cross breeding between O, P, and S? Okay, this is called mono. hybrid, cross. I'm only looking at the color of the flower. I don't care about the height and whatsoever. State the Mendel law. This is Mendel's first law. Okay, so what is this law? This is the law of segregation. Simple. Okay, so I'll leave question 3 and 4 to you to do. Okay, since we're running out of time here. Okay, 3 and 4. Okay, for 5, you can try. Okay, you can try question 5. Okay, please don't do this. Okay, this blood group will go into that next week. Okay, so that's it for this week. I hope you learned something new today. Okay, I wish to stress this again. This will definitely come out in SPM. So please put your time here. I would rather you not focus on chapter 4. Okay, a lot of people tend to focus on chapter 4. That's not as important as you think. Focus on this. This is much more important. Okay, so I hope this video is helpful. We will learn about blood group the next week. Okay, so again, if this video is helpful, you may give this a thumbs up or subscribe. Okay, if you're interested in joining our live class, kindly go to TTC Education on Facebook. Okay, again, I'll see you next week. Stay safe and bye-bye.